Thank you so much. I am delighted to be here. Thanks for tuning in to my talk. A little bit of background about some of the cells that I study and that I'll be talking about today. So for the last 7 years or so, I've been working on a subset of the immune compartment called T cells. So these are a really important player in adaptive immunity, and they're made up of numerous what we call plastic subsets. And so this feature of plasticity is really important. It allows cells to respond to a changing environment. So when you think about your immune system, this is of course very important. And sometimes we observe what we call intermediate cells, so cells that can adopt several phenotypes, or that of what is intermediate between two terminally differentiated or supposedly terminally differentiated cells. For example, we observe cells between a Th1 and a Th17 phenotype. And so this is interesting because whilst these cells are associated with protective functions, they're also affiliated with disease development. So for example, in rheumatoid arthritis or inflammatory bowel disease, we see an increase in the incidence of these cells. Understanding why these cells are adopting other phenotypes and characterizing them deeply is really important to offer insights into the immune system and of course disease development. And there's lots of different ways we can study and characterize these cells. And one of the tools that we use is looking at the epigenome. So I always like to define epigenetics because it does mean different things to different people. And so broadly, epigenetics refers to heritable changes in gene expression without alterations in a DNA sequence. So this is heritability referring to cell division. And there's lots of different types of epigenetic modifications, and the one that we study and that I'm going to talk about today is DNA methylation. And so this is a process by which methyl groups are attached to cytosines. There are other types of modified bases, of course, for example, hydroxymethylation. And so this is where we observe hydroxymethylated groups attached to cytosines. And this is important to note because DNA methylation and hydroxymethylation can have opposing functions in gene activity. And so what we've described is that when we observe methylation in certain regulatory regions, in certain cellular contexts, it's correlated with gene silencing, whilst the opposite can be true of hydroxymethylation. When we observe hydroxy in enhancers, for example, it's highly correlated with gene activity. And this opposing function becomes important when we talk about how we detect these modified bases. When we look at traditional methods like bisulfite, it's unable to distinguish the difference between a methylated cytosine and a hydroxymethylated cytosine. And this can lead to a muddy picture of the methylome. When we're trying to understand insights in gene activity, identity, and cellular memory, which is imperative for the immune system, this becomes paramount. And so that's why we became really interested in nanopore sequencing, because we could potentially detect multiple modified bases simultaneously without PCR, without bisulfite. We're characterizing DNA modifications directly on native DNA. And the concept of being able to detect any base by just training new models, we thought was really exciting. So the first question we had was, what does the DNA methylation profile of T cells look like with nanopore? And to study that, we developed a Cas9 targeted sequencing method. And so this method, we looked at in vitro polarized T cells, where we isolate naive T cells from mice and push them towards different fates in vitro. And so we targeted 10 regions, starting with about 3 micrograms of DNA, which is quite a lot. And we looked at important regions for T cell identity. We employed double cutting and barcoded 4 samples on one flow cell. And we did all of our analysis locally. If you haven't heard of Cas9 targeted sequencing, you first block available ends of DNA, and then you enrich in specific regions of interest with Cas9 guided RMPs. So it requires a lengthy library prep to enrich in your regions prior to sequencing. With this method, we obtain full gene length reads, so between 10 and 25 kilobases, which is a huge region to be able to enrich in and obtain a full gene length read. We also observe a beautiful enrichment in all of our target loci. However, shorter regions got a much better enrichment than our larger regions. For example, IL-4, we had a huge enrichment compared to integrin beta 8, which we observed a much lower enrichment. So that's an important consideration when developing this method. When we looked at the methylation profile, it was what we had hoped to observe. So DNA methylation in different subsets was clearly able to distinguish these different T cell subsets. So they had unique methylation profiles. Within just 10 regions of interest, we could clearly identify the different subsets, including a simulated intermediate cell subset called TH17s with no TGF-beta. Now, hydroxymethylation. We were very excited that the latest Dorado model was able to detect hydroxymethylation and promised to do so. We've been hoping to detect hydroxy with nanopore for a very long time, and we've tried many ways and failed. So Dorado was very exciting for us. And so with this, we wanted to validate this approach, and so we looked at central nervous system tissue. So central nervous system tissue has a higher level of hydroxymethylation. And so we isolated forebrain and adipose tissue from mice and looked at 5MC, 5HMC and detected that with Dorado. So we aggregated hydroxymethylation in enhancer regions, and we observed a clear enrichment in our brain enhancers. And while this is by no means a comprehensive validation process, we were confident we were seeing enrichment, and we were excited to look at what was going on in T cells. Unfortunately, hydroxymethylation in our regions of interest did not add discriminatory capacity for T cells. We're looking at differentially methylated loci in this graph, and we did not find any differentially methylated regions that we could aggregate these to. We think the reason for that is we didn't take a large enough flanking region either side of our promoters for our regions of interest. And if we had done so, perhaps we would have seen a more specific hydroxymethylation profile for each cell subset. I want to also mention using your off-target reads. So with Cas9, you generate around 1 gigabase of off-target reads per run. And we wanted to use that to understand what was going in our cells a little bit more. And to do that, we used a package called Mira to aggregate methylation in transcription factor binding sites. So please have a look at our paper if you're interested in doing so yourselves. So the next question we had was, do pathogenic T cells have a specific epigenetic phenotype? And when you're working with clinical samples, you're unable to have micrograms of DNA. So our previous method was not appropriate. So to study these more clinically relevant samples, we used an adaptive sampling technique. So in this model, we have a mouse model whereby we can track if T cells have changed their phenotype. And these T cells are affiliated with inflammatory bowel disease. And so to study these cells, we developed an adaptive sampling workflow to target 139 regions that are important for T cell identity based on our single sequencing data. So if you haven't seen adaptive sampling, it's whereby you can enrich for regions of interest during your sequencing run if they align to a specific reference or not. DNA is accepted or rejected during the sequencing, so there's no specific library prep required. With this method, we saw a clear enrichment in regions when we compared to random regions in the same run. When we also compared to whole genome sequencing approaches with the same overall sequencing depth, we also saw a clear enrichment. It was between 20 and 30x enrichment in our regions of interest, so it was a very nice approach. And of course, we saw a clear epiphenotype within our cells of interest. If you're interested in this, please have a read of our paper. So in conclusion, T cell subsets, they harbor unique methylation profiles, and whole genome sequencing is not necessarily required to study these different cells and discriminate intermediate cell transitions. Cas9 targeting will allow you to obtain whole gene length reads, but it requires high DNA concentrations. Adaptive sampling will also be able to enrich in your regions of interest, and it's more relevant if you have low starting materials. However, for both techniques, it's really important to take care when designing your regions of interest to ensure they're similar lengths. Hydroxymethylation within our target regions was not interesting or able to distinguish T cell subsets. And we really need to take this further and validate it more deeply to understand its role in T cells. I want to thank you all for your attention. Thank you so much for tuning into my talk today. I want to thank my collaborators and all the people that worked on these studies, as well as our funders, of course.